Hello friends, welcome back to my YouTube channel Nastin Padasala. In this video, I'm going to share with you list of important topics from which questions will be asked for GATE 2023 examination. The two, if you are applied for biochemistry paper under GATE Excel means, then this particular video will be definitely helpful. If you are prepared well means, you can use this particular list as a checklist. If you miss to study any topic, you can definitely study. Or else, if you didn't even start the preparation, if your preparation level is 0, you can use this particular list so that prepare notes only for these list of topics. It hardly has 60 topics only. So first study cells of immune system. Next have a brief understanding about antibody structure and type of antibody, function and the diversity. Next you should know about activation of B and T cells along with the development of T and B cells, and know about the DNA polymerase and RNA polymerase and what are the different type of DNA polymerase and what are the different type of RNA polymerase both in prokaryotes and eukaryotes. Next you should know about the translational inhibitor of ETC and uncouplers of ETC. Next study about the formula of retention factor, which is very easy of course, but many of you don't know, so just study the formula and work out few sums related to that particular thing. Next study about energetics of almost all biochemical pathway that has been listed in the syllabus. So you should know how many ATP is produced, how many NADH is produced, like that you should know about the basic information. Next study about the B-Ramber's law, and you should know about separation techniques. So only they will be asking the application. So separation techniques or some other molecular technique they will be asking only in the match the following only. So if you know the application alone, it will be very much helpful for you to clear the examination. Next, know about this isotope switching, self-tolerance. So these are fancy words from immunology topic. So again, I'm telling you, you can refer vocabulary section. So you should know the definition of those things, okay? Because generally in immunology general, these fancy words they'll be asking in match the following only, okay? Next, study about the purine and pyrimidine structure. Next, know about this transcription initiation site or transcription start sites both in prokaryotes and eukaryotes. Next, study about the enzyme and the coenzyme and the cofactor that are responsible for the catalytic function. Next, you should know about a complete details about all biochemical pathways starting from the person who discover the biochemical pathway till the energetics. At the same time, you should know about the regulatory steps or rate limiting steps and the enzyme which is catalyzing that particular regulating step. Next, you should know about protein folding. At the same time, you should know about list of disease caused by protein misfolding. Next, study about the classification of lipid along with the biosynthesis of lipid, and study about fatty acid oxidation, and know the entire structure of transfer RNA. You should know entire structure means you should know who discovered history of transfer RNA, and you should know about the biogenesis, how the tRNA is being generated and structure of tRNA, and also you should know about amino acylation of tRNA. Next, study about the differential centrifugation, which is again an important topic because each organelle will be separated by this particular centrifugation technique. So you should know about the centrifugal speed. At the same time, you should know the time point. Okay. Next, study about autoimmune disease, the self-antigen. Okay. And you should know about PCR because these time PCR related questions are asking repeatedly. Next, study about DNA microarray, ELISA and immunohistochemistry. These things are important. So first, if you are preparing for CSIR examination, you should study top to bottom. Since it is a GATE examination, you should know ELISA means for what purpose ELISA being employed, and DNA microarray means for what purpose DNA microarray being used. Okay. Next, you should know about biosynthesis of nucleic acid. Next, you should know how hemoglobin will be carrying both oxygen and CO2. Next, study about DNA binding motifs, the structure of transcription factor, and you should know about enzymes. So from enzymes, you can expect one question. So you should know about the enzymatic reaction, enzyme regulation, enzyme inhibition, and Michaelis-Menten equation, and also you should know about LV plot. Next, study about this major histocompatibility complex, MHC, class 1 and class 2. So in the QB book, you can be able to find a difference between class 1 and class 2. So at least study that particular single tabulator column. Next, you should know about the transport of molecule, order of molecule that are crossing this particular phospholipid bilayer. Next, you should know about how many ATPs are being reduced during aerobic respiration and anaerobic respiration. So see the difference between aerobic and anaerobic respiration. Next, you should know about glycosylation of protein. Next, hypersensitivity reaction. So there are 4 types of hypersensitivity reaction. Again, I'm telling you, in the QB book, you can be able to find a difference between all the 4 types. So you can study only that particular tabulator column along with the other things that you should know. So next, you should know about protein elution conditions in different type of chromatography. Next, study about this HMP shunt and location of biochemical pathway, where each biochemical pathway will be located in each organ. So you should know about the organelle and biochemical pathway, and you should know about epimer, anomer, and STS page and glycosidic linkage, blood group, antibodies, and study about O2 dissociation curve, and know about Sobel hypothesis and Abbay's equation, mutation, types of DNA mutation, study about structure of amino acid and the function of amino acid. So simply if you type amino acid notes for competitive examination in a single website itself, you can gather many information. So you can refer those important things. Okay. So next thing with regarding to Ramachandran plot and drugs that inhibit various biochemical pathway and glycogen storage diseases. So here I have given you in a yellow color thing. So you should study about each and everything. Okay. So you should know about lysozyme, ribonuclease A, carboxypeptidase, chymotrypsin. Definitely one questions will be asked from this particular thing. So don't leave this particular the thing that I had shaded in yellow color. Okay. Next know about the neurotransmitter and study about DNA damage and the DNA repair and the disease that are caused when there is a defect in the DNA repair system. At last, if you have time, and you should also study about the monoclonal antibodies like hybridoma technology and their application. So these are the list of things. So try to cover at least the topics which I have listed over here. Still, you have lots of time. Still, you have 10 to 15 days for your GATE Excel paper. So if you revise only the topics which I had listed over here, you will definitely clear the GATE exam. Thank you friends. Thanks for watching this video.